Hi, I'm Charles Wykoff, and it's an absolute pleasure and privilege to be here with a good friend and colleague across the retina space, Jason Ehrlich, who is MD, PhD, and currently serves as the Chief Medical Officer and Chief Development Officer of Kodiak Sciences. And today we're going to put Jason on the spot for two major themes. First, we're going to talk about his role and his experiences at Kodiak Sciences, where they are today and where they might be headed tomorrow. And then secondly, we'll dive into his personal story. What brought him from ophthalmology into the pharmaceutical side of industry and what that journey has been like. So Jason, great to be here with you today. Thanks for joining us.
Hey, thanks, Charlie, and thanks for having me. I really appreciate the opportunity, and it's a exciting, new venue and opportunity for people to talk about how they have, gotten, into the clinical trial or clinical drug development space. From where we all started training as ophthalmologists, and it's been certainly a really interesting and rewarding career journey for me so far. So appreciate the opportunity to get to share some perspectives and insights.
Awesome. So let's jump right in. So Jason, I love one of Kodiak Sciences' stated missions on your website, which says, quote, "Challenge the status quo." So tell me what that means.
Yeah, well, I think at the end of the day, everybody wants to do something that's impactful and important, right? We at Kodiak, our vision, is not to make Me Too medicines. We want to do things that are really going to advance patient care in clinically important ways, right? So that's really what we mean by challenging the status quo. If at the end of the day our medicine looks the same as the existing medicines, then that's not what we're trying to accomplish, right? But that's just, I'd say, really on the medicine's development part. But we also want to think about challenging the status quo in terms of how drugs are developed, right? For instance, at Kodiak, we're taking this approach with our lead medicine, the KSI 301. Long-acting anti-VEGF, where we're developing the medicine in wet AMD, in DME, in RVO, and in non-proliferative DR, all at the same time with a whole suite of pivotal studies, as opposed to doing it sequentially, which is the way it's been done in the past. One of the, and we can get a little bit further into it, but one of the potential advantages of that is a streamlined regulatory strategy where working with FDA, we negotiated to do, 2 studies in the lead indication and then 1 each in the subsequent indications. So assuming those studies are successful, that could form part of a pivotal package, right? So that's really challenging the status quo. How do you, rather than just proposing a traditional regulatory approach, how do you take advantage of all of the knowledge that has accumulated in the field, all of the experiences on the part of everybody who's done drug development over the last 5, 10, 20 years, and sort of stand on the shoulders of that, if you will, and then try to, further advance what's possible with the regulators, right?
I love that accelerated approach. I think you guys have really brought some insight into the space when it comes to how efficiently can we move the development process forward for the betterment of the field. And we'll get into KSI 301, but the one other basic question I would ask before we get there is, tell us about this science that you guys have, this body of knowledge that you have around ABCs, or antibody biopolymer conjugates, because this is new to the space of retina. And tell us about the science, and are there any other places in medicine where ABCs are being actively used in the clinic?
Right. Yeah, so the antibody biopolymer conjugate technology was invented at Kodiak. Victor Proroth, who's our co-founder and CEO, he, along with the other co-founder, really nurtured the exploration of the biochemistry and advanced it over really many years, in the background from 2009 to probably like 2015, 16, before it really started getting ready for the clinic. And the concept really started with the unique biochemistry and biophysics of phosphorylcholine. So phosphorylcholine is part of the phospho part of the phospholipid bilayer on all of your cells. It's a very biocompatible material, and it structures water. It binds water irreversibly in a really interesting way. And there was, Victor came across some of the scientific concepts around this when he was actually doing venture capital and like an entrepreneurial in residence opportunity back in 2008. And the science was around how phosphorylcholine is very biocompatible and stable. And then the question was, if you could make polymers of phosphorylcholine, how could that be useful? And there was a concepts around half-life extension. That's kind of where it started. And if you could hook up polymers of phosphorylcholine to large molecules or small molecules, could you develop drugs and have them be, could there be like relevant therapeutic differences? So, that then led to exploration of where in the body, these phosphorylcholine biopolymers could be useful as when attached to a drug, like a biologic and antibody. And the eye was a really compelling one because, if you think back to 2008, 2009, 2010, this was really still early on in the days of anti-VEGF. And, it was already clear at that point that, the medicines that we have, which are, that we had then, which are the same ones we have now, Lucentis and Eylea and off-label Avastin, don't last that long in the eye. And there had already been many efforts to come up with technologies and strategies to extend the duration of effect, none of which had worked at the time, right? The eye was a logical place where there was a very clear need for medicines lasting longer. Antibodies given systemically have a very long half-life, so you don't really even need to extend the half-life of something that's given like intravenously. But if you think about spaces like the CSF, the eye, joints, those are areas where a biologic or a macromolecule injected into that space will ordinarily leave relatively quickly. So those were areas that were really interesting. And then the company spent many years optimizing the design of these polymers. So there's essentially, it's like a 9-arm polymer, and there's all this custom synthesis that's done around an initiator, sort of that looks like a hand and a finger that then the polymer grows off of. Kodiak really pioneered the technology of polymerizing phosphorylcholine in a controlled manner. It's called controlled living polymerization. That's the chemical technique to result in a very uniform length of the polymers, and then took advantage of a lot of chemistry that's known about how to hook antibodies up to other things. So like antibody drug conjugates that are used in oncology, we use a similar chemistry to hook up the antibody with the biopolymer. So, at the end, basically what's done is we have in-house protein engineering. So we make our own antibodies, and then they all have like certain, the polymer is attached just at a certain site on it in a site-specific way. And then you have this large 9-arm polymer. So our KSI 301 anti-VEGF ABC is 950 kilodaltons molecular weight, right? So it's a very, very large molecule. And the phosphorylcholine is not just dumb mass. It doesn't just make it big. It turns out the biophysics and the biochemistry of phosphorylcholine, it does a number of interesting things that are really useful when you think about a medicine that you want to have last a long time in the eye. So it penetrates the retina very effectively in preclinical data, even more effectively than an unconjugated biologic, which is really very interesting. It kind of noodles its way through because it's not a big ball. It's sort of like 9 arms of spaghetti.
And is it integrating into the cell membranes in the retina, or is it staying separate?
It's probably staying separate. I guess the answer is probably not entirely known, but we would assume it's staying separate. Okay. Because it's just the phospho part of the phospholipid bilayer. It's not the lipid part.
Yeah, and some of your preclinical models on half-life extension compared to our current anti-VEGFs are pretty impressive. They're rather striking and very consistent with what we're seeing in the phase 1B. Give me the short answer, the preclinical extension of half-life. How much longer is this drug going to last, do you think?
Right. Well, I think, the preclinical models, I'd say suggest that it could last, maybe 3.5 times longer than, the existing medicines. But that's, maybe just like a kind of an approximation of, you have like a lot of different models to come up to like, well, how long does Lucentis or Eylea last in the eye? We probably last like 3 times longer than that. But, that also depends on like, well, where you measure. Are you measuring in the front part of the eye? You can't measure what the concentration of the drug is in the tissue.
Yeah.
But what we estimate is that relative to Eylea, if you inject people with the current therapeutic dose of Eylea and our test dose of KSI-301, at 3 months later, you have a 1,000-fold higher concentration of KSI-301 on average, right? There's a lot of variability between patients with all of these medicines, how long they last in the eye, and then how long the clinical effect lasts. But so part of it is the half-life extension. Part of it is improved access to the tissue. Part of it is very nice biocompatibility and preclinical models of safety, and it has relative to its size, rapid systemic clearance. Probably, not definitively known, but probably because we impair the binding systemically of the antibody to the FcRn recycling receptor because of where the polymer is conjugated.
Okay.
But at the end of the day, in terms of the eye, when you inject a biologic into the eye, it sort of it diffuses out through the tissues. So a very large molecule is going to take longer to diffuse out. The unique thing maybe there about phosphocholine is there had always been a question, if you make it so big, it won't work anymore. But clearly we're having a very potent anti-VEGF effect. So either that hypothesis was wrong, or the unique way in which we have tissue access is allowing the medicine to work despite its large size.
So let's talk about that. You're in the midst of a large ongoing phase 1b program across sort of the 3 most common exudative retinal diseases. Very promising data so far to date that's still unfolding. Where do you see KSI fitting? Where do we go from here? You have this accelerated timeline to get to clinic. So from your perspective, what is the date that we as a field can look forward to where it's potentially in the clinic? And then where do you see this fitting with our current anti-VEGF agents?
Yeah, so, I think 2 parts there, right? So in terms of the development plan, we have an ongoing pivotal study in treatment-naive wet AMD. That study is called DAZZLE. And that one, we're hoping to finish recruiting by the end of this year. Right now it's, about mid-September. So at the end of this year, we should hopefully finish recruiting. And then we're, just in the process of launching some phase 3 studies, pivotal studies in treatment-naive DME and RVO. So we would expect, if those recruit according to, typical timelines, and there's a lot atypical in the world right now, as you know, we would expect to have top-line results from that whole suite of programs, really starting from the early part of 2022 on through the middle part of 2022. And then, you have to bring all of those data along with manufacturing together into a licensing application, which we, if those studies are successful, we would, hope to try to file towards the latter part of 2022, which could mean it could be potentially approved sometime in 2023.
Remarkably
which
fast.
in the scheme of drug development is like right around the corner.
Yeah, remarkably fast turnaround from your first patient in. So what's down the road for KSI-301 beyond KSI-301? What's in your pipeline? I think you have a lot of opportunities with this ABC platform. What are you looking at next?
Yeah, so now that we've really have started to generate clinical data and start to understand how the medicine behaves, the safety, what the durability is, we really want to think about further expansion of the pipeline in a couple of interesting ways. The first is our next medicine that's lined up is called KSI-501. It's an anti-VEGF IL-6 dual inhibitor. There's some really very interesting data on the role of IL-6 in diabetic eye disease, in wet AMD, in uveitis, uveitic macular edema. So there's a number of places in which IL-6 inhibition on top of VEGF inhibition could be beneficial. It has to be tested in the clinic. So what we would anticipate there is, once that medicine goes through all the IND enabling work, some interesting translational medicine studies where we would look at it in a variety of conditions and start to see where it may be more useful than anti-VEGF monotherapy.
And then simplistically, is that 2 antibodies that you're attaching to this biopolymer, or is it a bispecific antibody? How are you getting 2 targets out of this?
That particular one is a trap antibody fusion. So it's a trap molecule that's fused to an antibody.
Okay.
And that's a pretty interesting set of technologies, protein engineering technologies. But conceptually, the platform itself, could be hooked up to any different type of biologic of interest. There's different formats for bispecific antibodies and that have been used, and our platform could be potentially used with a variety of those formats.
So you
The other
mentioned,
really
good, go ahead, please.
Oh, yeah. The other really interesting thing that we're doing a lot of work on, I'd say in the background, is what we call triplet inhibitors, where we combine a biologic, like a dual acting biologic, with a polymer that is decorated with hundreds of copies of a small molecule drug. So one important thing that I didn't mention earlier is that our antibody biopolymer conjugate is stable in the eye. It doesn't fall apart. So it's not like it's not a degrading polymer. It doesn't leave residues in the eye.
Right.
It doesn't dissociate in the eye, which is, I think, important both for durability but also for safety. And what another technology that we've developed is essentially you can dope the polymerization of the phosphorylcholine polymer with a different monomer that has a chemical linker to a small molecule drug. And that small molecule could fall off in a controlled, releasable way. So what that could allow you to do is essentially target 3 different biologies at the same time. We haven't revealed or disclosed what targets we're going to be inhibiting or targeting with these triplet inhibitors, but we're really excited about those for dry AMD, for the sort of retinal component of glaucoma, the neuropathic part of glaucoma, a number of different areas where the efficacy may be substantially improved or there may be like any efficacy with by inhibiting multiple biological pathways. Dry AMD, glaucoma, wet AMD, these are complex multifactorial diseases, and it may be to get the best benefit, you need to inhibit or target multiple pathways. The cool thing about having a small molecule is it allows you to target both extracellular and intracellular targets.
So you're mentioning everything in ophthalmology, but before you did mention sort of CNS diseases, you mentioned orthopedic diseases. Are you guys looking outside of ophthalmology also, or so far we eye focused?
We are really focused on retina at Kodiak. We think that there's a benefit to focus.
Yeah.
There's right now, there's no mid-size ophthalmology or retina-focused pharmaceutical company, retina-focused pharma company anymore. If you think about the companies of the past, like Allergan and Alcon, when they were really primarily eye-focused, really the only there's very few freestanding ophtha biotech and pharma companies anymore. Mostly they've been acquired. So we think there's actually like an important need for that in the community as well. I'd say the only mid-size ophtha company that's freestanding right now is Aerie, in terms of having like marketed products. And they've been focused really on the front of the eye. So we think there's a lot of value to focus in retina in particular. We think there's a lot to learn. The manufacturing for retina is complex. We think there's a lot of value there. Not to say that these other areas couldn't potentially be interesting, but it's not something that we're really focusing on right now.
So then last question about Kodiak, because I want to talk about your personal story, which is fascinating in and of itself. I would be remiss not to bring up safety. You've touched on it briefly a couple of times. The field is in the midst of sort of a lot of different challenging safety issues with many products in development, and we're still learning what all this means, these inflammatory responses with multiple different products. Are you guys seeing any safety signals that we should think about?
Right, great question. And of course, safety is important. It's always been important, but certainly there's been a bit of a renewed focus on intraocular safety in the community. When you think about, over the longer arc of time, over the last 5 or 10 years, interestingly, a lot of the focus was on, systemic safety. There are differences in the medicine systemically, and that's not really a current topic of discussion now. So, so turning to KSI 301, what we've seen so far, we've given just about 550 doses of KSI 301 in the open label phase 1 study. And the safety profile there, we've been very pleased with what we've seen. There have been only 2 cases of trace to 1+ vitreous cells, both of which resolved with no sequelae. So those were quite mild, no vasculitis or retinitis or anything unusual. So it looks right now tracking very much similar to the safety profile of Lucentis and Eylea, and we're very glad to see that. Obviously, a lot of attention to detail on manufacturing, quality parameters, and so on. But ultimately, you need to test the different medicines in the clinic. We have a variety of novel technology platforms that have been evaluated in the clinic over the last several years, some of which have been well tolerated and some of which, we've seen have not been well tolerated. We think that the fact that we're using phosphorylcholine, which is this very biocompatible material, and we're using fully humanized, native form, full-length antibodies, both of those are important factors that may benefit us in terms of the safety profile. But again, tracking very similar to the Lucentis and Eylea profile from what we've seen so far, and in the masked DAZZLE data as well from that ongoing study based on the last IDMC review, for instance.
One other question, just to dovetail off what you just said, Jason, the antibody, clarify for us, it's a different antibody, a different sort of epitope that's being bound, or is it the same epitope that's being bound by other commercially available anti-VEGFs?
Right, it's the same epitope that binds on VEGF as Lucentis and Avastin, right? So it binds to the same spot. It has, we'd say like very good affinity and potency when you measure the potency and the affinity of the antibodies in a variety of different in vitro systems, right? So it's actually really cool. It turns out that with the addition of the phosphorylcholine biopolymer, you actually get better potency measures than with an unconjugated antibody. And we think that's because the sort of structured water environment of the phosphorylcholine polymer favors the, it favors the hydrophilic antibody ligand interactions, and it disfavors sort of the background hydrophobic interactions that can occur and that can contribute to sort of background noise in the system. So we actually have better potency than with an unconjugated antibody, which is pretty nice.
Very promising. Look forward to a lot more data over the months and years to come. But I do want to pivot and now talk about your personal story, right? You have an incredible CV, Jason, just to be blunt, right? Undergraduate at Princeton, medical school at Stanford, residency at Stanford in ophthalmology. You did a 1-year clinical research fellowship, I think, at Genentech. You were then on staff at Stanford and the associated VA. And then you spent almost a decade at Genentech, where you rose through the ranks, it looks like, and eventually was global head of clinical ophthalmology, and then transitioned pretty close to 2 years ago now, September 2018, to Kodiak. So clearly you've trained at premier institutions. You've had some incredible contributions to major scientific advancements in our space over the last 10 years. And my question for you is really, what has inspired you along the way, and what drives you on a day-to-day and a longer-term basis?
Yeah. Well, I think it's to me, really, it's largely about patience, right? When I was chief resident at Stanford, this was, right when I think we had like the first or second Pascal laser, pattern scanning laser in the United States, because that technology was developed at Stanford. And we had that at the county hospital. And I was doing Pascal panretinal laser on lots of patients with proliferative DR. This was, Avastin wasn't even available at the county hospital at that point. So, thinking there's got to be more things that we can do for patients, and that was like very striking for me personally. And I think it really impacted my career in a lot of ways. And then I did MD PhD at Stanford. Before that, I was always interested in sort of the nexus of science and clinical medicine and drug development. There were a lot of faculty members at Stanford who were involved in biomedical device companies, involved in either founding them or consulting for them, and also for biotech companies. The Bay Area with, Genentech, really, one of the first biotech companies ever, was between Stanford, Genentech, and UCSF. There was like a tremendous amount of entrepreneurial activity in the sciences in biotech. And so as a student, you got to sort of be part of that, or, see it in different ways.
So talk to me about that transition specifically. You're a clinical ophthalmologist, you're on staff at a great institution. What sort of led to that transition into industry? What was the impetus for that?
Yeah, well, I'd say in part it was fortuitous, and in part it was planned, I suppose. I finished residency in 2008, and I was looking for a fellowship, more training to do for a year. My wife is an immunologist. She's on the faculty now at University of Texas. She was a postdoc at Stanford. I was a resident there. And it was the 2008, like the economy had collapsed and all of the faculty positions that she was searching for had all those searches got halted. So she was still at Stanford. I was looking for something to do for a year. And Emmett Cunningham, who you probably know, who's been involved in Ophthalmology Innovation Summit and is a notable ophthalmics investor now. He was out in the Bay Area, and he volunteered at Stanford, and he knew the Genentech people. He said, "Hey, Jason, you should think about this fellowship." And Stanford didn't have a lot of clinical fellowships in ophthalmology at that time. I met the Genentech people. I learned about this clinical research fellowship, and I had always been interested in clinical trials. So I said, I'm going to go. They offered me the opportunity to spend a year with them. I took that opportunity, and it was really transformative for me. I learned about how clinical trials work, statistics, regulatory, safety, protocols, drug development strategy. As a research fellow, I was kind of like, really in that with the team who was developing Lucentis at the time. And I really fell in love with that, and it sort of reconfirmed that, for me, I could really have a big impact on patients by doing drug development, running clinical trials. And, coming back to my experience at the county hospital in San Jose with diabetic eye disease, the first program that I ran at Genentech was a phase 3 studies for Lucentis and DME. They had already been ongoing before I got there, and then I had an opportunity to take those over as the medical lead. And, that program ultimately resulted in the first medicine being approved, the first intravitreal medicine being approved for diabetic eye disease in the United States. We had an FDA advisory committee. It ended up, it was a complex regulatory package because there was nothing approved for, as an intravitreal therapy at the time. So, that set of experiences was really transformative for me. It really, when I thought about once that medicine was approved for Lucentis was approved for DME, how many thousands or tens of thousands of patients benefited in the first 6 months that drug was available. So that's the kind of impact that you can have.
Yeah. So for your colleagues out there that are currently in clinical ophthalmology, but interested in sort of what you've described, this whole pharmaceutical development pathway or device development pathway, and they're interested in looking at industry positions, or they're already looking at them, what sort of advice do you have for clinicians that are interested in making that transition at least part-time? We've seen a lot of sort of part-time transitions, or full-time.
So I think, Charlie, my advice to people, it depends a lot in part on what kind of career do they currently have, and what stage in their career are they, right? So, the advice would be probably different for somebody who's just finishing up fellowship, the clinical fellowship, as opposed to someone who's established academic faculty or, established in private practice. I'd say at a high level, if you're just finishing up a fellowship or early in your career, I think that's a good time for people to pause and consider, is there other training that they might want to have? And, I thought, for instance, the Genentech fellowship that I participated in, which is something that they, I believe they still offer, those are the types of programs that can be really impactful because, you can really learn a lot without, in a structured environment, with a plan that really helps you understand, would this career transition or move really be right for me, right? And there's a number of those fellowships. Some of them are more academic, research oriented, but the Genentech fellowship is at least one of the more notable industry ones. Not the only one. I think J&J and Merck and other big pharma companies have offered these, although not necessarily specifically in ophthalmology. Now, some people also, after a fellowship, decide that they may want to pursue like a different type of additional degree, like an MBA, that could be useful either for, in industry or for, business management of a large group practice or a hospital or things like that. And, I think that could be also useful. Maybe a little bit less so if you really want to do drug development, but if you want to do banking, or if you want to do investments, if you want to do more of the commercial side of biotech and pharma, that type of degree, the block and tackling and the networks that you make can be really important too. So those are things to think about if you're early in your career. What I would also say is, and we can maybe come back to this, is whatever stage in your career you're at, it's really important to have really good mentors. People who can give you advice, can be a neutral sounding board, because these are hard decisions, especially if you've spent, many years training to do clinical practice, and then you find yourself in a position where you say, well, is that still what I want to do, or is that going to give me the most satisfaction, allow me to help the most people in the way that I want to? Those are hard decisions and transitions, and it's often really useful to develop relationships and cultivate relationships and keep relationships with mentors from residency, from medical school, from earlier on in your career. Those mentorships can be really, I'd say, important in helping you think through these transitions.
So let me dig on that for a second, because I couldn't agree more. Mentors are so important to my development as who I am and sort of most of us. But how do you find a mentor, right? For people that say, yeah, I need a good mentor. What are they supposed to do, nuts and bolts? And then maybe tell us a little bit about the key mentors that you've had over the years. Yeah.
Well, Atul Gawande, who's a clinician, who's a surgeon who writes a lot for the New Yorker, wrote a great article in the New Yorker many years ago about having a coach, and how, everybody should have a coach regardless of what stage in their career they're at, that he got a coach, even though he was, one of the, best endocrine surgeons in Boston or whatever. I've had a number of important mentors over, my career so far. I think when I was a medical student, when I was a resident in ophthalmology, folks like Mark Blumenkrantz and Emmett Cunningham were important mentors for me. Kuldev Singh, also on the faculty at Stanford. Especially as I was considering a career that was either more research, clinical research, or sort of entrepreneurial drug development oriented. Just being able to speak with those people, they all had different points of view. And never really asking for like, well, tell me what I should do, but just understanding their history, their stories, and advice that they would give. How could I explore a career in industry? How could I, who should I talk to? Bob Kim, who was at Genentech, he was an important mentor of mine. As I grew into industry, I cultivated relationships with a number of the, more senior leaders at Genentech. Folks that probably not many of your listeners would be familiar with, people who, knew the ins and outs of that organization, could help me understand how to navigate as a clinician successfully in it. And then over time, I built a number of relationships with other, more senior clinicians, not necessarily ophtha people, who were maybe like 10 years ahead of me in their career, at Roche, at Novartis, and also in the, in the investment community, in the VC community, people who I had met over time. And again, it's to me, it's not really a question of like, well, tell me what I should be doing, but more, having somebody who can really be a sounding board, who may have been in these, had similar experiences to yours, and can say, I'm trying to decide, should I, stay in this industry job, or should I've been offered an opportunity somewhere else? Or, I'm struggling in this large political complex environment of this big company. And I'm afraid to talk to somebody within the company because it may look bad for me. Those kinds of things. Those relationships that you have, which you can develop by going to a variety of meetings, like the innovation summits, the industry-focused meetings in my case, or, in the case of many people in retina at ASRS, at the Retina Society, Macular Society, AAO. It's always really valuable to I think, develop, meaningful relationships with people who are, 5, 10, 15 years more senior than you in addition to people who are contemporaries, just because they, the benefit of like the wisdom and maturity is always really useful.
Jason, fascinating personal story. Thank you for those insights into your own journey. Loved hearing about Kodiak. Maybe now the last chapter to add on here. You're a chief development officer and a chief medical officer. Tell us what those phrases mean and what are sort of the key leadership roles across industry that the space needs to be aware of?
Yeah, great question. So, at a smaller company, and Kodiak is definitely still smaller, the chief medical or chief development officer kind of means you do everything. I'm involved in building the team, hiring in the different functions, clinical operations, regulatory, clinical science, statistics, pharmacology. Some of those are roles that you may have to identify consultants for. Some of them are roles that you hire your own people and you build those teams over time. So that's a large part of my time. Another large part of it is obviously overseeing all of the clinical development of our medicines. And that involves, like the day-to-day things, what's going on in this study, what's happening with a particular patient, just working with the teams to make sure that, everybody's on the same page about patient management, for instance. But it also runs all the way over to making sure that the contracts with all of our different vendors and partners are what we want them to be and that we're building the right relationships with those folks. It's also, keeping abreast of the science, working with an R&D organization, working with clinicians, understanding what are the needs in the field, staying up to date with all of the competitive information and all of the latest developments, so that, you can make sure that your medicines are, you're developing in the right areas, you're pointed in the right direction, and you're following the science. It's also, I also get heavily involved in all of our regulatory interactions and all of our regulatory documents, protocols, regulatory meetings, European trial applications, scientific advice, pediatric plans. That's just like a smattering of the things. I also provide like IT support and cheerleading and a shoulder.
Man with many different hats.
So, I think that's the job of, senior leaders at a smaller company. To be honest, it's also the job of, senior leaders in pharma too. It's not that different. Just, some of the details are different because at a big company, you have a whole large team that takes care of things like procurement, but, you still as the senior leader in ophthalmology at a big company, you need to be, understand all the details of what's going on and be accountable for those things. I think that's really important to people's success. Yeah. And then, another thing that a CMO does, which I think is probably different than what mid and senior level leaders typically would do of Ophtha would do in a pharma company, is a lot of interactions with investors, with investment analysts, with investment bankers, lawyers, as well as board members, right? So, in many ways, there's more stakeholders, external stakeholders in a small company. And that's part of the learning experience of being in a small company, is how to interact with all of those different people. Because it's, there's a number of different considerations of, how you finance a smaller company as opposed to the ongoing investment in R&D that occurs as a sort of a matter of course in a big company. So that part's quite different, and it's been really fun for me to learn about that.
Jason, thank you. Incredible insights. Really appreciated hearing your story. Good luck in the years ahead. Look forward to continuing the conversation.
Appreciate it. Thanks for having me today.